Hello, students. Today I'm going to give you an online lecture as we begin Module 4, which is all about DNA and RNA, as well as genetics and heredity. This first portion is going to be just about 25 slides, and then I will give you instructions onto what type of test or assessment I want you to complete to let me know that you've done it. So let's get started. All right, so you may have heard of the term genetics before, and genetics is an area in science that really means the study of genes and how traits can be passed along from one person to the next. So you might have frequently seen this picture on the left, which let me just give myself a pointer so it's going to be a little bit easier to see. You may have seen the picture on the left, and this is going to be your genetic material, which we've talked about before. Remember, that's one of the characteristics of life. And this is one of America's favorite families, just wanting to show how genes can be passed along from parents to children. So you may have heard of this term before, and this is studying the genes, which I'll give you a definition of what that is. And also, once we understand those genes, how we can move the genes from one place to the next and pass them along. So as we know, all living organisms are going to use DNA as genetic material because all living organisms have genetic material, but some viruses can use RNA. So we haven't really discussed viruses too much, but viruses kind of have this debate on whether they're living or not. Regardless, they can use RNA, but if something is living, it's going to typically use DNA as its genetic materials. This is going to include all of the items here. So of course, plants and animals, bacteria, fungi, proteins, archaea, anything that we've discussed so far, except for viruses, which can use RNA. So an example test question is you're looking under a microscope and you realize you're looking at the norovirus. What type of genetic material is a possibility for this virus to have? Could it be A, DNA, B, RNA, C, both RNA and DNA, or D, none of the above? And it will be both because they can use RNA, but they also can use DNA as well. All right, so if we're wanting to look at our DNA structure, we're first going to start off at the very, very small portion here. So this is going to be our nucleotides. So the first question here is, what macromolecule is DNA? And I hope you said it's a nucleic acid, because it definitely is. So the first part of the DNA structure is going to be our nucleotide. So think of your nucleotide as like your Legos. These are going to be your little tiny building blocks that we can use to make DNA or RNA. So this is going to be the monomer of a DNA or RNA strand. Remember, mono means one, so the single individual unit is a nucleotide. And we'll do some examples of those if you don't remember ones we've already covered. Once you get a lot of nucleotides together, they begin to build up a string, or what we called a strand. So a strand of DNA is just going to be a linear polymer. So that just basically means a straight line of either DNA or RNA. And the strand, it's really important to know that the strands are linked together by covalent bonds. So what this is saying is that the nucleotides, so when we start talking about those A, T, G, G, C, C, all of that stuff, the bond between them is a covalent bond. And if you remember, covalent bonds are the strongest bonds because that's where you share the electrons. So we want that strand to be the same. We don't want to copy our RNA and we end up losing bits and pieces because it's not glued together very tightly. We want it to be glued together tightly, so we use covalent bonds in between the nucleotides or in between the strands. Once you get two strands together, they start to form what we call a double helix, which is two strands of that the DNA together, and they start to kind of form a spiral. So you can see that here in this picture to the right, where the strands are connected, but they're starting to spin. So thinking of like a curl or some type of coil where it begins to spin, that's what we called a double helix. And the bonds that are between the two strands, so if you're looking here, the little links between the two strands, that's actually a hydrogen bond. So if you remember, hydrogen bonds are the weakest, and this is important because we're eventually going to pull our strands apart. So as you see here, you have two individual strands that are tied together by covalent bonds because they're strong. We don't want to lose our message, but we split them apart in the middle, and the bond that's in the middle is a hydrogen bond because it's going to be a lot easier to pull apart. Finally, once you have a set of double helix together, begins to coil. We'll talk about what it wraps around. But once it forms this X-shaped structure, that is called a chromosome. So that's going to be your DNA, which is your double helix and your strands and nucleotides interacting with different proteins, which I'll tell you about, and that compacts it and makes it really tight and very, very close together. That's going to kind of form this X-shaped chromosome that we're so used to seeing. And finally, if you're thinking of humans or plants or animals, once you're looking at the complete set of chromosomes or the complete set of genes they have in their whole body, that is going to be called your genome. So it is the complete set of genetic material within an organism. And if you're thinking about a genome, what other organelle have we talked about that contains genetic material? So if you didn't know, the DNA is housed in the nucleus. And what other organelle in our body for humans, or for animals at least, have genetic material? The answer is the mitochondria. So when you're talking about genome, you're also including the mitochondria as well. That's just another interesting, important fact. All right, so again, our nucleus contains our genetic material, and this is just showing the backwards way. So of course, when we look at it, it's going to be in the chromosome form. If you break that chromosome apart, you're going to see the double helix. That's where you get the spiral shapes. And then as you zoom in on it a little bit closer, you can see the blue strands, which are going to be held together by covalent bonds. And then you can see the nucleotides in the middle that are represented by the different colors, and they are going to be held together by hydrogen bonds. So this is just showing an up close and personal view of the genetic material. All right, so like I said, our chromosomes, you see this here, our chromosome is going to be a key factor in helping to keep our DNA very, very tight. So a little fast fact is that if you took one chromosome from your body, from a little teeny tiny cell that you have to look under a microscope to see, and you expanded the chromosome, the DNA can actually stretch up to 3 feet. So even though the DNA is really, really tiny inside that cell, it's because it's been coiled up and wrapped up so tightly. If you stretch it out, it would be about 3 feet. So we've done a good job of compressing that and putting it really tight together around those proteins. And in humans, we have 23 pairs. So please keep in mind, 23 pairs of chromosomes mean we actually have 46 chromosomes total, but they're a pair, right? So think about a pair of socks. If you're looking at 3 pairs of socks, you technically have 6 socks because a pair represents 2. So we have 23 pairs of chromosomes, and we actually have 46 chromosomes total. When you're looking at the structure, you can see, again, it's an X-shaped structure. Each arm of the X, we called it a chromatid. So you see that's labeled here and also labeled here. And in the middle of the chromosome, we call that area a centromere. So just like centra is kind of like central right in the middle, a centromere is the middle of the chromosome. All right, so let's look into the nucleic acid monomer and try to understand what these monomers are made up of. We've actually covered this when we did macromolecules, so this may be a breeze. It's exactly the same slides that I used back then, but this is just a refresher if you're not sure. So again, a nucleotide is made up of 3 components. The first component is a phosphate group. That's here, that's listed here. It's a P group that you see, as well as some oxygens present. You then have a 5-carbon sugar. If it's 5 carbon, we call it a pentose, because that just pentose means 5. And you see it's a carbohydrate because there's an OSE at the end. And that 5-carbon sugar can be different depending on what type of nucleic acid. We'll get to it. And then it has a nitrogen containing base, or sometimes we call it a nitrogenous base. So the 3 parts that make it up are a phosphate group, a 5-carbon sugar, and a nitrogen containing base. So let's look into each one. When we're talking about that 5-carbon sugar, it can either be ribose or deoxyribose. So ribose is going to be present in RNA, ribonucleic acid, and then deoxyribose is going to be present in DNA, deoxyribonucleic acid. With our nitrogen containing bases, we have one set, which this might be new information for you. One set are our purines. So purines have a double ring. So what you see here in this image is a purine because it contains two rings. And the structure or the specific nucleic acids that are purines are adenine and guanine, so A and G. A way some students remember this is the saying pure as gold. So pure as gold. Purines are pure as gold, right? Because you think of, you want gold to be pure, right? So that's a good way for people to remember the names of the purines. The other one sounds like a pyramid. It's a pyramid, but these are single rings. So it's just one ring. Again, with your purines, if you're talking about gold, you probably want two gold rings, right? Instead of one, pure as gold. But with our pyramids, these are just single rings. And the pyramids we have are thymine, cytosine, and uracil, which we'll talk about when we see uracil, which is actually only in RNA. So this might be flip card things to remember, or some type of question where it says name one purine, name one pyramine, and that's what you got there. All right, so again, with our 5-carbon sugar that they have to have, ribose is here. It has oxygen on that carbon number 2, and deoxyribose on carbon number 2. You see it's without oxygen, so deoxy without oxygen, it's just an H there. That's the difference. Our nitrogenous bases, you can see them here. Again, our purines, because they have two rings, is adenine and guanine. And then the pyramines are the uracil, thymine, and cytosine below. Again, this is just another image showing that, and more particularly showing the pairing between the bases, where A always goes with T and G always goes with C. And this is the same thing in RNA, but in this case, A only binds with U and G still continues to bind with C. So again, showing you our structure for DNA. A is always going to bind with our T's, G is always bind with our C's, A, T, G, C. And then what it's showing here is you're having the 5-carbon sugar, which in this case is deoxyribose, with the phosphate group that's linked to another 5-carbon sugar linked to a phosphate group. It's in this direction. And then we'll talk about in a second how, if you notice, it's actually upside down on this side because this side is anti-parallel. But I'll cover that in a second. And then you see the same thing with RNA, except for you do not have any T's. They're replacing the T's with the U's. So again, as we're looking at the structure, we want to make sure we understand how to name our carbohydrates. So when we name our carbohydrates, we actually have carbons 1, 2, 3, 4, and 5 present. It's important to know that on carbon 1, that's where your nitrogenous base is connected. So remember, this is a pentose, meaning there are 5 carbons. So if you count them, it's carbon 1, 2, 3, 4, and 5 kind of sticks up. The peak of the house is the oxygen. Excuse me. And when you're counting your carbons, the first carbon, you want to make sure that base is attached to it. If you had to have a question, what carbon is the base usually attached to? It's carbon 1. And the phosphate group is attached to carbon 5. So everything in the middle is just works with the sugar, but carbon 2 is where you determine if it's ribose or deoxyribose. Carbon 1 is where your base is attached. Carbon 5 is where your phosphate group is attached. So that could be a really easy, simple question, whether it's a matching question or fill in the blank question or true false question, making sure you kind of know those 3 numbers, especially carbon 1, base, carbon 2, type of sugar, and then carbon 5, phosphate group. All right, so we've looked at the structure a few times. This is the same slide that you had from the macromolecules, but I'm reiterating it. So when we link together our nucleotides, they're going to be linked by covalent bonds. And that strand that forms the backbone of our DNA, we call it a sugar phosphate backbone. That is because I showed in the picture before, you have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. That's basically the strand. There's going to be some nucleotides that stick out in the middle, but the strand itself is called the sugar phosphate backbone. DNA always runs in 5' to 3'. What does that mean? That means the 5 carbon, right? Do you remember the 5 carbons where the phosphate group is? It's going to be the first carbon. It's going to be carbon, the very top of the strand, for instance, and it's going to run down from the 5 prime side down to the 3 prime side. If you remember, your carbon number 3 is right here, right? If you're looking at my arrow, because our base is connected to carbon 1, carbon 2 tells us it's what type of sugar it is, and this is carbon 3. So it goes from 5 on this end down to 3. So 5 to 3. Then again, 5 to 3, 5 to 3. It's always running 5 to 3 prime. Now, DNA runs in an anti-parallel direction, meaning if one side of the DNA runs from 5 to 3, the other side is going to be from 5 to 3, but it's going to be flipped. So it's anti-parallel. So normally parallel would be this side would be 5 prime, the bottom side would be 3 prime. If it was parallel, that means the same exact direction. This top side will be 5, this bottom side will be 3, but it's anti-parallel. So you have to basically flip one side. That's because they wouldn't pair up the right way. So one side of your DNA is going to be 5 prime that goes down to 3 prime. The other side, although it still runs in the 5 prime, 3 prime direction, the 5 prime starts at the bottom and it goes up to the 3 prime. So if you were replicating this DNA, on the left side, we will be replicating down because that's 5 prime to 3 prime. And on the right side, we will be replicating up because we still have to go from the 5 prime to 3 prime end, but they're just flipped. And I can explain this a little bit more in class, but I want you to kind of try to understand it as much as you can ahead of time so you have the whole week to practice. All right, so the bond that connects our phosphate groups is called a phosphodiester bond. Phosphodiester bond. So that is going to be the bond that that is going to connect the phosphate groups that we have. Again, like I mentioned, the hydrogen bond is going to connect those nitrogenous bases, and that's going to be between each one. You see those little dashed lines? That means it's going to, they can easily be broken. And finally, A binds with T or U if we're talking about RNA, and G always binds with C. Again, this is what I mentioned before. On this side, you see it's the 5 prime that's going down to the 3 prime end. And this is just showing one of those sides in which you have the 5 prime carbon here. If you can see my pointer right here, the 5 prime end is going to be at the top and it's going to go down to 3 prime end, 5 prime down to 3 prime end, 5 prime down to 3 prime end. All right, so we're switching gears a little bit and talking about some famous scientists. I'm going to cover them really briefly so we don't need to discuss a lot of information about them, but I did want to showcase what we have so far. All right, the first one is a woman named Rosalind Franklin, and she was pivotal in understanding the first structure of DNA. So she did this thing called DNA distractions, which you don't have to know about, but it's basically where you shine light through wet DNA and then it kind of projects like a shadow onto a screen. But she was the first person to kind of see this X format. So especially for a woman in science back in these days, it wasn't very common. So we want to acknowledge Franklin for being the person to come up with the first DNA structure. The second person we want to acknowledge is Chargaff, and he was the person to realize that A always binds with T and G always binds with C. What he noticed is that once he looked at the base percentages between various animals, he always saw very similar numbers between adenines and thymines and between guanines and cytosines. So for example, in humans, if you looked at our base pairs, about 30% of them are adenine, but also 30% of them are thymine. So it makes sense for them to pair together as compared to 19% are guanine and 19% are cytosine. So it makes sense for them to pair together. So Chargaff is the binding rule. Franklin is the first DNA structure. And then finally, you probably heard of these fellows before, Watson and Crick. They are credited for determining structure. This has been really controversial before because a lot of individuals think they kind of had some help from other people, but they didn't really those people didn't get the credit. But still, for the credit of who determined DNA structure, it is Watson and Crick. So that's who you need to remember for that. All right, so in the 1950s, once we determined the structure of DNA and the components, we really were trying to understand how DNA is replicated. So there were three different models that were proposed. We're only going to study one, and I'm going to tell you about it on the next slide. But there were a lot of different thoughts about what could happen. One thing we do want to keep in mind is that whenever we replicate DNA, we always call our main strand, the original strand, the parental strand or the template strand. So imagine if you had to write a copy of something from me. You're going to have some type of template or some type of original copy, and you want to copy it exactly the way it is. So when we start talking about the replication, we always want to keep in mind our what we call parent or template strand, because that's going to kind of be your original strand that you use to make a copy of your new strand from, which we will call a daughter strand, and I'll introduce that in a bit. So what we determined is that DNA replicates on a semi-conservative replication. So semi means half, and conservative means very strict. So it's kind of strict, but there's a little bit of wiggle room. So what happens first is the two parent strands will separate, and they will serve as kind of a template or a guide for how to make the daughter strands. So you see the parent strands are in red, and the daughter strands are going to be in blue. They're going to first separate. So imagine you having a red line up here and a red line down there. The new nucleotides that come in that form the daughter strand have to obey this ATGC rule. If not, it's going to be a problem. You're going to start making a different strand that you intend. But because of that, and because we know this ATGC rule does not fail us, you're going to end up with two strands that have the same we're in this first round. You're going to end up with two strands that have the same exact genetic material as the first strand. So imagine the parent strand here is ATT. The bottom strand will be TAA. So if we took our ATT and moved it up here, the new daughter strand follows that same rule and it's TAA. So it's going to be the same type of format that you have here. I'm sorry I can't write on this online lecture, but I can definitely help you all if you're having trouble. So semi-conservative is very simple. It just basically means that our parent strand will separate and it will serve as a template to produce the daughter strand. That's what that means. All right, so this is an even better image of that. You have your parent strand here. It separates, you break those those hydrogen bonds, and then you're going to form new daughter strands, which are indicated in blue, to match up with that parent strand. And you see, if you're looking across AT, AT, or TA, TA, GC, GC, they're going to always be the same because of that A and T and G and C rule. All right, so this is going to wrap up the first online lecture. This covered our genetics intro, and there will be an additional one on mitosis and chromosome structure.